Here we go. Let's try this. All right, so 5.2b is we're going to use chi-square to determine if cells are dividing normally. Because remember, what cancer is loss of control of cell division. Cells are at an abnormal rate of division. So what we do in this lab is we take a chemical, and that chemical is called lectin, and it's a protein. You can tell it's an IN. And we take some cells and we expose them to this lectin. And basically what we're going to decide is this chemical carcinogenic? Can it cause cells to divide abnormally? Because that's what cancer is. So can this chemical cause cells to divide normally? Is it carcinogenic? Well, if we want to see if cells are dividing normally, we have to look at cells that are dividing normally first and compare them with these cells that have been exposed to the chemical. And that's what this picture here is. It says that these are cells that have only been exposed to water during this time. These are the normal cells, and they're going to show us normal rate of division. And then we can use their information to compare it to these cells that have been exposed. So let's just talk about, in general, what percent of these cells should not be actively dividing. And so we're going to look for cells that are in interphase or cells that are in geophase. And basically, you can tell them because you can't see the DNA in them. Because remember, DNA is in chromatin form when a cell isn't actively dividing, and you can't actually see that DNA under a microscope. You can only see DNA whenever a cell hits prophase. The DNA coils up into chromosomes, which you can then see with the microscope, okay? So when I'm looking at these cells, that's interphase, 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 okay? But the cells that you can see DNA and it kind of looks like worms, you're looking at a cell that's in a stage of mitosis. So I would probably call that one prophase. This one would be a good anaphase because it looks like they just got freshly pulled apart. Metaphase is kind of hard to point out, but I would say that these X's are kind of lined up in the middle here. Here'd be a metaphase. I'd probably call these prophases. This would be a good telophase. See how they're way over here on the edge, and it even looks like a cell plate is starting to form in the middle, okay? But all in all, if I count the number of cells that are in a stage of division, I came up with 9. So I'm putting over there. And cells that are not in an active stage, I counted 31. So if we figure out the percent of the cells that are in these two stages, again, part divided by whole, 31 divided by 40, there's 40 cells total that I analyzed. And again, these are normal cells. When I do the math, that gives me 0.78 for this one, times it by 100 to get to a percent. And so 78% of the cells should be in interphase if these cells are dividing normally. And 22% should be in some stage of mitosis. Okay, again, this is our normal rates of division. Here's the normal. We need something to compare these chemically treated cells to see if they're dividing normally or abnormally. Okay. All right, so here's where our chi-square comes in. So these are the cells we've exposed, and we count them, and we find 366. So these are your chemically treated cells. 366 were in interphase, and 150 of them were in mitosis. The total number of cells that we analyzed, it looks like 516 cells exposed to chemical. Okay. And I'm going to go and put my variables here. So they were in interphase or geophase. You can't really tell from looking at them. And then these were in a stage of division, one of the stages of mitosis. Remember, according to our formula, we have observed and we have expected. Remember, in the expected column is where we put percents. Well, that's handy. I just calculated percents. So the expected is the normal division. If these 516 cells are dividing normally, then we would expect 78% of them to be an interphase. We would expect 22% of them to be in a stage of division. Remember, we write our percents in the corner there. And then how do we get our numbers here for the expected? Like I would take 0.78 times it by what?
Observed.
And it's going to be that, the observed total. I like to draw my arrows, so then you guys are always like, oh yeah, she always draws arrows on this. I try to do it the same way each time. So 0.78 times 516 gives us 402 of these 516 should be an interphase if they're dividing normally. And then 0.22 times 516 should give me that number. I should see 114 in a stage of mitosis if they're dividing normally. And how can I tell that I'm doing that column correct, that I did it correct? Abby,
It should,
go ahead,
it should
Adam.
add up.
Yep, they should add up, and they do. So everything checks out, okay? So I'm good to proceed on here. So the formula says O minus E squared, O minus E squared divided by expected, and then I'm going to add those numbers up, and I'm going to get that chi-square. Okay, so the chi-square is going to tell us whether we accept or reject our null. So let's again just review what a null says. Null means none. If you get an annulment, it means that no marriage happened. So null means none. We always say there is no significant difference, and I'm going to abbreviate because we have a lot to do, between the observed, which was 366 in interphase, and 150 in mitosis. And what is expected? Which is 402 in interphase and 114 in mitosis. I'm going to put a little caret mark in here because I feel like I need to say something else because I did this in my other two classes. And what is expected if cells are dividing normally? So if the cells are dividing normally, we're expecting 402 in interphase and 114 in mitosis, okay? So how many of you think 366 and 150 are a good fit to 402 and 114? So you're going to give me a thumbs up or a thumbs down here in a second. Because that's what we do in a chi-square. We're trying to figure out if these two numbers are close to those two numbers. So what do you guys think? Thumbs up if you think they're a good fit. Thumbs down if you think that they're a poor fit. See those votes. Okay, well, when we all do this, or I have half of you here and half of you here, it means that that's why we need chi-square, because we all have to decide the same way. Let's do our math here. I've got it already worked out for us, so we can go faster. It's 36 and 36. If you get a negative value, no big deal, because we're squaring it, and that's going to disappear. It's 1296 and 1296. And then just to make sure you understand what I'm doing, you would do 1296 divided by 402. And that gives us 3.22. And then down here, you use 1296 divided by the expected is 114. And that's the one that gives us a high value. All right, we add those up. 14.62. That's our chi-square. All right. So we then have to figure out what to do with this number. We need our chart. Remember, this is on the formula sheet too. We always use a p-value of 0.05, and then we have to know which degree of freedom to use. So see, it says degrees of freedom, 1, 2, 3, 4, 5. Those are the degrees of freedom. For the degrees of freedom, it's always n minus 1. N is our number of variables, which is 2. So 2 minus 1 equals 1. So there's our degree of freedom. And here is our critical value. Okay, so 3.84 is called a critical value. I'm just going to put CV for critical value. Sometimes when you guys are justifying your decisions, you'll tell me 3.84 is the degrees of freedom, and that's incorrect. You're going to lose your point. Okay, the degrees of freedom is 1. My critical value is 3.84. All right, so now we've got these numbers, we compare them. And we can see that our chi-square value of 14 is way over that critical value. So that means you reject the null, okay? The null says that these two sets are a good fit. The chemically treated cells are a good fit to the normal dividing cells, okay? That's what our null says. And we just rejected that, okay? So if we reject, then that means that these two are a poor fit to one another. So is the chemical affecting cell division? You bet it is. Because it's saying that these numbers are a bad fit to the normally dividing numbers for those chemically treated cells. Okay. Now the justify part is justify the rejection. Why did you do that? And that's where you guys have to make sure you can explain that since our chi-square value was greater than the critical value of 3.84, and you need to tell me where that comes from. You have to explain that you know how to use the chart. So if you say and explain that 3.84 comes from using that p-value of 0.05 and determining or calculating the degree of freedom was 1, that's how you got to that 3.84. So when our chi-square value is greater, you reject the null. Okay, so you got to make sure you mention it's greater or less than, how you got that critical value, that you know how to use this chart. You have to prove to your grader you know what you're doing. Okay, so that means our answer is yes, that chemical is causing those cells to divide abnormally. All right, so let's review a little bit and let's talk about those checkpoints. So you can just, you don't have to get this out, but actually you do need to get this out. So here is our, pull this up. I pulled out 5.1 here. And we want to add something. Okay. So remember that there's 3 checkpoints and you have to know the checkpoints are between different stages and be able to identify that for me. Okay. So let's talk about this first checkpoint, is that the cell is checking its DNA for mutations. Now, to get a visual of what that looks like, remember every job in a cell is done by a protein for the most part. And that's what this protein is doing. So you have proteins that will check the DNA for damage, for mismatched base pairs. Sometimes sections of DNA are completely missing. And when that protein finds something that's not right about the DNA, it'll bind to the DNA, and then it will activate other proteins to help repair it. And that's one thing that we said needs to happen at this checkpoint. If the DNA is damaged, then hopefully it can be repaired by enzymes. And if it can be repaired, then the cell should go on to S phase. Okay, so this is what we mean by the DNA is being checked and it's being repaired. Okay, now if the cell cannot repair the DNA, it should exit the cell cycle, and that's called apoptosis. We say, well, sorry, apoptosis is a cell should die. And what that looks like, if we really want to watch cells dying here. Okay, so just watch kind of carefully the whole of the screen, and you're going to start to see some changes take place here in these cells. Sometimes I think students think that apoptosis is cells literally just like blow up, like you stuffed a piece of dynamite inside of them. And it takes time. Again, it's just digestive enzymes will start to break down all the molecules of the cell. And so then you just have little pieces of the cell that are being produced here through apoptosis. So that's what apoptosis looks like. okay? Now, according to the lecture, I said have it memorized. Name a protein that triggers apoptosis. What is the name of a protein that signals apoptosis? It starts with a P. Right, write it down. Okay. So the protein that activates apoptosis is called P53. And unfortunately, in a lot of cancers, the reason why someone has a cancer cell is because their P53 protein is not working as it should. If a cell gets mutations, P53 should be activated and the cell should initiate apoptosis, okay? In cancer cells, their P53 lots of times isn't working. So let me show you, this is what a normal karyotype, a karyotype is just a picture of human chromosomes. So we see 46 chromosomes here, okay? So this is the DNA out of a normal cell. This is the DNA out of a human cancer cell. Isn't that crazy? Like, look at all, there's extra chromosomes, broken chromosomes, there's missing chromosomes, and there should be some P53 getting activated, and the cell that has this DNA should die, but it doesn't. And it keeps dividing and dividing and dividing. So P53 is an important one for you to note. Anytime a cell activates this P53, that cell should die, all right? So P53 is not a bad molecule. P53 is a good molecule. And let me get to this quick, okay? And as a matter of fact, P53, along with lots of different proteins, have to do their job so a cell can divide when it should divide or die when it should die, okay? So expect plenty of questions that look like this, because all cancer is when cells lose control of the cell cycle because one or more of their proteins stop working. So for example, let's say that someone has ovarian cancer. It could be that this one protein right here isn't made correctly. So that ovarian cell divides out of control. But what's so difficult about cancer is if AKT was causing cancer in one person and another person that has ovarian cancer, their AKT is fine, but their SOS protein is messed up. So cancer treatment now and in the future is individual based. We have to learn what's wrong in that person's cancer cells, what proteins are not working correctly, so then we can figure out a treatment for that person. So cancer is very individualized. Cancer treatment's very individualized and will become even more so as time goes on, okay? So this is the reason why we keep showing you guys signal transduction pathways, is because that's the future to fixing, preventing, and treating cancers. You have to understand what's wrong with the person's signal transduction pathway. So it's when our proteins aren't working as they should to control the cell cycle or initiate apoptosis. All right, so let's talk about reading these. If you see an arrow, you have to understand that that means that when that, like P13K, when it's active, it turns on AKT, okay? When you see something like you see here in red, I like to think of that looking like this. So if I hold my hand out to you, you come up to me and I go, stop, like this. So think of this being the hand here at the end of my arm. That means that this molecule, ditamine, stops AKT, will not let it do its job, okay? And again, all these proteins are important. They all have to work correctly though. And if they don't, you can end up with a cancer cell. All right, so let's practice. Here's a scenario. You guys are gonna give me a thumbs up or a thumbs down. And so I need your cameras on so I can make sure that you're with me. If AKT is activated, will the cell divide or not? Get that answer in your head. If AKT is activated, will the cell divide or not? If AKT is activated, will the cell divide or not? So if you think division will happen, thumbs up. If you think division won't, thumbs down. Where's all my people? About half of you turned off. Okay, so it will divide because that's the job of AKT. It says AKT causes proliferation. You need to know that word. Proliferation means cell division. Proliferate means to become more in abundance. Cells will divide. Okay, so you got to know your symbols. So let's do some more practice. Let's grab 5.2 here. All right. Again, cancer. When good proteins go bad. Okay. So the first question, we're just going to start the first question and see if you can get two proteins named here. It says, according to the diagram above, name two proteins that act like accelerators of the cell cycle. So if a protein is an accelerator, it doesn't mean it's good or bad. It just means it causes division. Okay, causes division, and we need cells to divide, so we need accelerators of the cell cycle. Okay, if we say that we have a protein and it stops the cell cycle, think of it as a brake. It prevents division from happening. And that's also a good thing because sometimes we need to stop cells from dividing. Okay, all right, so let's just focus on number one. See if you can name two molecules from these diagrams that promote cell division, that cause cell division. All right, I'm going to call on some of you to tell me what you thought here, and I can guarantee I'm going to call on people that have their cameras off first.
Can I volunteer?
Yeah, go for it, Alex.
This is MDM2.
Okay. MDM2?
Because it looks like it's inhibiting P53, and that leads to inhibiting apoptosis.
Okay. So if you have P53 memorized as a break of the cell cycle, that makes this question really easy. Okay. So P53 is a break, and breaks are important. Okay. So what is something that takes out P53? Because that will be a molecule that promotes cell division. So what's something that stops P53? I would agree. MDM2 does. What's another one?
The HMGA, is that an I?
Who knows? I think it's a 1. Okay. So if they're active, the cell is going to divide because P53 is going to be inhibited. Okay. What's another molecule that assists with P53 or turns on P53? Because that's like the foot on the brake. You need two things. You need the foot and you need the brake.
HIPK2.
Very good. Okay. All right. So what should normally happen if we have DNA, I'm just going to draw some DNA here, and there is a mutation, then what should happen in cells is that mutation activates proteins which will activate HIPK2. And when HIPK2 is active, it activates P53, and that P53 will cause apoptosis. Okay. Can anyone guess what that little popsicle thing is? Did HIPK2 give P53 a popsicle? And then it's all active, like children? Any idea?
It's like a signaling messenger. Is it like a hormone or a protein?
That's a good guess. But do you remember this? Oops, ADP. So again, what we use to activate molecules, we put a phosphate group on them. So that's a phosphate group, just so you kind of get used to seeing that. And that comes from ATP. Okay. All right, let's kind of review a little bit. We're going to go look at number 8. And this is why vocabulary and knowing the events of the cell cycle, like what we put here, is necessary to answer questions about like cancer cells like we see here, okay? And this is just chock full of vocabulary. It's important that we can picture events so that whenever they're describing events, we get an idea of what's going on, okay? All right, so we're going to kind of draw some of these out. It says cancer cells result from a variety of different mutational events. Which of the following is least likely to result in the initiation of a cancerous tumor? So least likely, okay? So what's going to lead to no cancer or no cell division? Okay, so if cell division happens, you could get a cancerous cell. But if something stops cell division, you're not going to get a tumor forming because that needs rapid cell division. Let's take a look at A. A is important. It says a receptor mutation results in the activation of a cell division pathway in the absence of the appropriate ligand. Woo! Okay, so there's a lot you had to know here for this one. I want to show you this video real quick because I think this brings some really important ideas. Remember, I've said several times that cells should not divide until they get a signal to do so. Okay,
And we into a person's bloodstream and attacks the.
And we talk about cell or cancer is due to mutations in the DNA. So let's make sure you understand how the DNA relates to proteins, relates to cancer. So this is DNA, and DNA codes for proteins. I don't know how many times I've said that, right? And so this DNA is coding for proteins like receptor proteins. So here's receptor proteins, and they have a job. It's their job to receive a signal, okay? Now, these receptor proteins are supposed to receive a signal so the cell knows when to divide. So here comes a signaling molecule. There's a ligand. We talked about ligands before. And ligands bind to the receptor, and that receptor protein should be sending signals to the cell to divide, to make proteins to help in cell division. I know my computer's lagging here, but I'm waiting because I want to show you one thing. So this is a good protein. It's a normally functioning protein. We need it to happen for normal growth and development. But watch what happens if there's a mutation in the DNA and this protein is made in the wrong shape. There's no ligand binding to that protein. Yet this shape is an active shape, which is activating molecules and activating the cell to divide, even though the cell is not getting a signal to do so, okay? So what I mean by, cancer, it's when good proteins go bad, or it's when mutations in a cell result in misformed proteins that are activating events that's going to lead to rapid cell division, okay? So going back to what it's saying here, it says a receptor mutation. So let's think about, here's a cell, we've got a receptor protein, and it's not been made correctly. It says an activation of a cell division pathway. So that means there's no ligand. It says absence of a ligand, but this is sending a message to divide, even though the cell should not be dividing. So would uncontrolled cell division happen? You bet. This would result in possibly cancer. Okay, so let's look at the next one. A mutation results in the loss of the ability to produce, I said memorize, p53 is a tumor suppressing protein. So we can't make p53. Okay, so p53 is a tumor suppressing protein. And how is it supposed to suppress tumors? It initiates cell death. Okay, so if there's cell death, there's no division. Okay, that's what p53 does. p53 does that. But the question says p53 isn't made. This can't happen. So will the cell divide? Yes, it will. That could result in a cancerous cell. C says a defect in the cell cycle checkpoint prevents a cell from entering the S phase. So let's kind of think about what that would look like. Remember, we drew the cell cycle. You were with me last class. And it looks a little something like this, G1, SG2, right? The 3 phases of interphase, okay? And this is what's it's saying, is a defect will not let the cell move into S phase. It stops, the cell stops. So is the cell going to divide? Right. If the cell stopped in the cell cycle, it means it doesn't go on to the next stage, which means it doesn't go on to the next stage, which means it doesn't actually go on to the division part, right? All right, we probably found our answer here. Because if no cell division is happening, you're not going to get a tumor. But let's just read D to make sure we understand this one. It says at the anaphase checkpoint, separation of chromatids occurs without all centromeres being attached to kinetic core microtubules from both poles. Oh, that's a lot, okay? So let's practice with some vocabulary. So it says that the sister chromatids are being separated, okay? So remember, sister chromatids, there's 2 of them in an X. It says they're being separated, right? And it says that they're being separated despite all of them being hooked up by spindle fibers to the poles. These aren't hooked up, right? So let's think about what would happen here. If this cell continues on, you're going to get these 2 sister chromatids pulled apart, these 2 sister chromatids pulled apart. That X is chilling right here. So if a cell splits into 2, look what we got. Extra chromosome. Remember those HeLa cells have like 200 chromosomes and they should have 46? Okay. So here's a cell that is dividing when it shouldn't. This is a checkpoint. This cell should have initiated apoptosis because not all of them were aligned in the middle and hooked on to kinetochores. So we've got some uncontrolled cell division happening. This could lead to cancer. Okay. All right. So you're going to be heading into breakout rooms. And I need you to practice with this material. So we didn't get a chance to work on the back of 5.1. That's where I want you guys to start today in your breakout rooms. So I want you guys to flip it over and go through and see if you guys can answer those questions together. Remember, it might help if one of you projects the screen in case not everyone has the worksheets. Whenever you get done with that, then I want you guys to then start working on 5.2. And the more you get done, the better, because we're going to move on tomorrow into lecture 3. And lecture 3 is all about DNA replication. So we learned that DNA replicates in S phase. Well, now we're actually going to see what that looks like according to the DNA, right? So the breakout rooms, I'm opening them up. I don't care which one you guys go into, but the more questions you guys get done, the better.